Welcome back for more AP Biology. We're going to be shifting gears today and going from big organisms, so dealing with circulation, going back to cells. In particular, we're going to talk about chromosomes. So we're going to deal with what is the genetic information inside of living cells. This is going to be our first part of this because eventually we're going to need to add on to it. There's only a little bit and text reference. So when we look at multicellular organisms, one of the things that we notice is we have lots of different types of cells, and each of those cells need to have the information necessary. So for us to have circulation, have all of those different body parts, or for plants to have circulation, have all the different plant parts, you need to make all those cells. So they need to be able to replicate, because we go from one cell to a whole bunch of cells. Now obviously something needs to transfer between each cell, some type of genetic information, some type of hereditary information. We learned this from Gregor Mendel, and then it's his further rediscovery of his work, namely because we used to think that you would take two little baby microhumans and then you'd fuse them together and voila, that made you. But Mendel's work said, no, it doesn't work like that. We actually have discrete units of information, and somehow they need to be inherited. That, of course, led to a whole bunch of questions like, well, what is that stuff? So a German in the 1800s was examining plants and as they replicated, and what he noticed was there were certain points during the replication that if you were to stain them, you would happen to notice these weird thread things that would show up. Eventually, he didn't do the naming, but eventually it became known or called that those little thread things were called chromosomes because they were colorful bodies, and these processes had names like mitosis and meiosis, which were named after the fact. But he noticed that, there are things that are being composed here, and he named those chromosomes, or the substance that made them up, called chromatin. He wasn't the first to characterize that chromosomes were there, but he certainly made it popular. Through lots of analysis by several groups of people that spanned a little while, what we've determined is those chromosomes, that substance, chromatin, is composed of three different chemicals: DNA, RNA, and protein. And the references to all that stuff is actually up at the top, so if you wanted to see where that information came from. It's actually quite fascinating because one of them is brand new. So I have, of the four authors there, I have three of the four. I don't have Adrian Rodriguez Campos. I couldn't find a picture of him, but they're the other three. So everything that they kept looking at was suggesting that we have genetic information bound inside of this chromatin stuff. And what we would notice is certain times you could actually see the chromatin in the chromosomes, and other times it was just a big old blob that we would just call the nucleus. And when they would condense down, when that chromatin would condense into actual shapes that we could notice, we started giving them names. So when you look at this, it's going to be vocabulary soup, I apologize. That entire X structure is called a chromosome. The middle part that connects them is called a centromere, meaning the center part. But each of those lines, so if you look at it, you have, of the X, you have a right side and a left side of the X. Each of those halves of the X is also called a chromosome. But also, when the two are touching each other, we also can call those two chromosomes that are connected at the centromere chromatids. The moment if I could split them apart, they are no longer chromatids, but they're still chromosomes, which gets all confusing, and they're made out of chromatin. If I look at that centromere, we have a short half and a longer half of each part of the chromosome, or sister chromatids. The short part is called the P arm, and the longer one is called the Q arm. It's just a whole bunch of vocabulary that we happen to use. It's not that it's super vital that you know those words. I just don't want to drop them and then have you say, I don't know what you just said. If we look at us, meaning a eukaryotic organism, what we notice is we seem to have duplicate sets of chromosomes, meaning I could have one complete set, and we number them by order of size. So chromosome number 1 is just the biggest, and chromosome for us 22 is the smallest. So that's just the way that they get ordered. It's very genius. What we notice is every organism or every type of organism will have sets of these things. So it's not that you will have, you could have a whole bunch of number ones but never have a number 2, 3, or a 4. It's you will always have a 1 through however many number that they have. If you have one set of all of them, we call that being haploid, or an N. If you have 2 of them, so you have 2 number 1s, 2 number 2s, 2 number 3s, we call that being diploid, or 2N. If you have 3 sets of all of them, we call it being triploid. If you have 4 sets, it's tetraploid, or pentaploid, or whatever. So we can just start multiplying them out. Is that a thing? Yeah. Some organisms seem to be okay having multiple sets, being 3N, 4N, 5N, 10N. Others seem to always be either N or 2N. So it kind of seemed to depend on the organism. If I were to look at what you have altogether, we call that a karyotype, which is just here are the contents of the nucleus. And if we look at us, humans, our karyotype has 46 chromosomes. So we have 23 different pairs of chromosomes. And what we notice is when we look at ours, the male and the female are different. There are some chromosomes that will be the same all the way throughout, and there will be some chromosomes that are different all the way throughout. So, for example, males have 1 through 22 the same, and then they have 2 weirdo chromosomes that don't seem to match any of the others. One of them we call the X, one of them we call the Y. If we look at a female, she will be 1 through 22, 2 sets, life is good, but she has 2 X's and she does not have a Y chromosome. There may be something to that. Who quite knows? What we do know is if we look at prokaryotic organisms, we don't see anything like that. We don't see chromatin, we don't see chromosomes, but we know there's some type of genetic information in there because we could test and find that yes, indeed, there is genetic information. So clearly they must operate on a mildly different level. The question then becomes, how do these things replicate, and what do those chromosome things have to do with it?